welcome back to my channel, and today in this video we are going to study about the Mendel's law of independent assortment. So to understand independent assortment, we have to work with two characters. When two characters are involved, that means two genes are involved because one gene is responsible for one character. So when two characters are involved, that means two genes are involved. Now here the cross we study is a dihybrid cross. Why? Because di means we are studying with two characters. It is a cross between hybrids of two character. Like monohybrid cross, that means it is a hybrid of one character, and when we are crossing over one monohybrid with another monohybrid, we call it as a monohybrid cross. Similarly, in dihybrid, we will cross two dihybrids. Dihybrid means the hybrids of two characters involving two genes. So here in independent assortment, or to study independent assortment, we have two genes. One gene which is responsible for the shape of the seed denoted by R. It has two shape, that is capital R, which is a dominant round shape, and small r is wrinkled recessive. Similarly, one more gene is involved for second character, and the character is the color of the seed. It also has two variations: yellow seed with capital Y genotype, that means it is dominant, and green seed which is small y, that means it is a recessive character. So now we have two parents, P1 and P2. P1 with a pure line, capital R, capital Y. That means the dominant characters. R for round and Y for yellow. P2, that is parent 2, is having the pure line of recessive character, that is small r, and small y, that is wrinkled and green. Here we are dealing with two characters. Remember, in monohybrid cross, we were dealing with one character. Here we are taking two characters together. So from P1, that is parent 1, we'll get the gamete capital R and capital Y. Similarly, from parent 2, we will get the gamete of small r and small y. Now, what will be the F1 generation then? Capital R, capital Y, small y. So, the offsprings are all phenotypically round and yellow, because though they have small r and small y also in their genotype, but as we studied, only the dominant gene will express. So, capital R is present, so the shape will be round. Capital Y is present, the color will be yellow. So all the F1 generation offsprings will be round and yellow with the genotype capital R, capital Y, small y. Remember again here why we are taking capital R, capital Y, small y, everything, because we are dealing with two characters. So R for one character, Y for another character. Now for the F2 generation, what we have to do? We have to cross the F1 generations. So we will cross two round yellow with genotype capital R, capital Y, small y. Now what F2 generation we will get? So these many four types of gametes in F2 generation we will get. What are those? Capital R, capital Y, capital R, small r, capital Y, small y. Similarly for parent 2 as well. So for both the parents, we'll get four varieties of gametes. So both dominant, then one dominant and other recessive, and both recessive. Now what we have to do? We have to make the offsprings. So these will be the offsprings. How? Capital R, capital Y, capital Y. So this is capital R, capital Y. Similarly, from here capital R, again this one, capital R, this is small y, again this one, capital Y. So capital R, capital Y, small y. For this, again from here only capital R, from here small r, capital Y. For this one and this one again, the first one, capital R, capital Y, small y. Now come to this one. So you have to add both these. So capital R, capital Y, small y. For this column, you have to add these two. So capital R, small y. So like this, you have to fill the columns. Now, after filling all the columns, what are the phenotypes? If this is capital R, capital Y, here it will be round and yellow. Here it will be round and yellow. This one will be also round and yellow. This one will be also round and yellow. This one again, round and yellow. This one, round and green. This one, round and yellow. Round and green. Round, yellow. Round, yellow. Wrinkled, yellow. Wrinkled, yellow. Round, yellow. Round, green. Wrinkled, yellow. Wrinkled, green. Got it? So now, what is the phenotypic ratio? How many we have round and yellow? 1, 2, 3, 4, 5, 6, 7, 8 and 9. So we have 9 round yellow. How many are round and green? So for round and green, we have 1, 2 and 3. How many we have wrinkled and yellow? 1, 2 and 3. And how many we have wrinkled and green? 1. So the phenotypic ratio for F2 generation in case of dihybrid cross is 9:3:3:1. Now, if the phenotypic ratio is 9:3:3:1, what we can note from this ratio? This 9 is for round and yellow. This middle 2, 3 and 3. 1 is for round and green. Another is for wrinkled and yellow. This one is for wrinkled and green. So if you remember, we started with parents P1 and P2. P1 was round and yellow. P2 was wrinkled and green. So in F2 generation, with respect to the phenotype, the maximum we obtained like parental phenotype. The minimum we obtained is also of parental phenotype, but which one? Which is the recessive character. But the middle two, what we got? We got hybrid of one dominant and one recessive. In this case, with round and green, round is dominant, but green is recessive. Here, wrinkled is recessive, but yellow is dominant. So, two ratios, 9 and 1, we obtained like parental phenotype, with the 9 belonging to the dominant parental type and 1 belonging to the recessive parental type. But the middle two, 3 and the 3, we got the hybrid of dominant and recessive. One character is dominant and the other character is recessive. Correct? So what is the interpretation? That means the characters are not linked. In the parent, what we had? R, R, Y, Y. Capital R, capital R, capital Y, capital Y. If this would have been linked together, then they would have behaved as a single gene or a single character. In that case, able to get this hybrid ratios. So that means the characters are not linked. They are separate characters because of the outcome of separate genes. The second interpretation is the maximum part of the ratio shows the parental dominant phenotype. The minimum part of the ratio shows the parental recessive character or phenotype. And the hybrid phenotypes are with equal intermediary ratios. Clear? Just now I explained you that two ratios of 9 and 1 is like parental type, with the 9 the dominant parental type and 1 with the recessive parental type. But the between 1, 3 and 3, these are the hybrids of two phenotypes having one dominant and the other one the recessive character. So with this ratio, the main interpretation is the characters are not linked. They are separate. If they would have been linked, then they would have behaved as a single character. Now that means what the law of independent assortment tells us. It tells us that the gene encoding different characters separate independently. Because they separate independently, because they are not linked, so it is not like that if in the parent round and yellow is found together, in the offspring they will always be together. It is not like that. Round can go with green also. Green can come with wrinkled also. Wrinkled can go with yellow also. So any characters can get connected with any other character during the segregation of gametes or segregation of genes specifically. So that means the law of independent assortment tells us that the gene encoding different characters separate independently. Each pair of alleles segregate independently from other pair of alleles during gamete formation. In case of round, in case of yellow, here wrinkled, here green, they are segregating independently. They are not connected with each other. Now, what is the requirement for law of independent assortment then? The gene should either be present on different chromosome, or if they are present on the same chromosome, they should be present very far apart. The minimum distance required for independent assortment is minimum of 50 cm, that is centimorgan. That is centimorgan. So minimum of this much distance between the genes are required for independent assortment. Otherwise, if the distance is very close enough, we will study further in genetics. Then in that case, the two characters or the two genes transmit one generation to next generation in a linked way. Those are known as linked genes because they always go to the next generation together only. They don't get segregated like this. So to get segregated like this, to get separated like this, there should be a distance between two genes. Either they should present on different chromosome, or if they are present on the same chromosome, they should be far apart. Now, independent assortment is the main reason for variations. Why I'm saying this? Human chromosome, how many? 23 pairs. That means a total of 46 chromosomes are present. Now, for one pair of chromosome, two gametes can form. For two pair, four gametes can form. For three pair, 8 different gametes can form. So that means the formula is 2 to the power n, where n is what? The pair of chromosome. For offspring, we need two parents. So from one parent, 2 to the power 23 different combinations or arrangements of gametes can be found. From second pair also, this much only the combination or arrangements of gamete can be found. Now when the offspring is produced, so in the child or in the offspring, 2 to the power 23 into 2 to the power 23, this much variations are possible. That means from single parent pair, these many variations of children can be produced. So that much variations of children can be produced from a single parent pair. So independent assortment is the main reason why we are different from one another. Why so much variations are present in a population. Why I don't look like my brother, or we don't look like each other. So because from a single parent, these much amount of variations are possible. So independent assortment is the main reason for variation in nature. Now, one question from my end for you all. What would be the phenotypic ratio in the F2 generation if round with yellow and wrinkled with green are linked together? Independent assortment, such ratio 9:3:1 is telling us that the genes are not linked. That's why we are getting this hybrid of one dominant, one recessive character. But what if they are linked together? In that case, what will be the phenotypic ratio? Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.